So Mendel, to get his second law of inheritance, the law of independent assortment, he used a dihybrid cross. So he's looking at two different characteristics at the same time. So a dihybrid cross is a cross between F1 dihybrids, so hybrids with two different characters that are transmitted to offspring, and he wanted to know were they transmitted as a package or independently. So here we can see a cartoon of this experiment. So we had true breeding parents, which had two particular characteristics. In this case, they could be yellow or green, and they could be round or wrinkled. So we had true breeding yellow round peas and true breeding green wrinkled peas, and when he crossed them together, just like when he crossed the true breeding purple and white flowered plants together, he got all plants that had yellow and round peas, or expressing both of the dominant phenotypes. So he was testing his hypothesis to see whether those two characteristics were somehow linked together. Would he get only yellow round and green wrinkled peas, or would he get all the permutations of yellowness and greenness and wrinkledness and roundness? And what he found was that he did get this. The evidence supported independent assortment. That the genes for these characteristics were transmitted to the offspring independently from each other. And he got this very characteristic 9:3 to 3:1 ratio based on how the gametes were assorted. And using that dihybrid cross, he was able to come up with his law of independent assortment, stating that for every pair of alleles that segregates, they're going to segregate independently from all other alleles at other loci. But this is only true if you are talking about genes that are on non-homologous chromosomes or vary apart from each other on homologous chromosomes, because genes that are physically linked to each other on the same chromosome tend to be inherited together. So what that means is that Mendel's traits that he was looking at were found on different chromosomes. So maybe the round wrinkled alleles for the genes at locus 1 are on chromosome 1, and maybe the yellow-green alleles are found on chromosome 7 at a different location. And with that knowledge, it makes sense that the independent assortment would occur. If the genes for roundness and wrinkledness and yellowness and greenness were found physically on the same chromosome, he would not have observed those familiar proportions of 9:3 to 3:1 in that F2 generation. So we don't always see a simple Mendelian inheritance pattern. Often, when we're looking at the genetics of characteristics, we don't see that it's quite as simple as what Mendel observed. And there are a number of reasons for that. For one thing in particular, genes don't necessarily only have two alleles. There can be more than two alleles for a particular gene. But even when we see more than multiple alleles, we find that there are some basic Mendelian inheritance patterns that we observe. So sometimes we see situations where we don't have this straightforward dominant recessive condition. We can have things like incomplete dominance or codominance. Sometimes we find that there are more than just two alleles for a particular gene. Or maybe we'll find that a gene may have, one gene may affect multiple aspects of phenotype. And we call that pleiotropy. So let's review each of these in turn. So the situation with the purple and white flowered pea plants was one of complete dominance, meaning the purple phenotype was completely dominant to the white phenotype. But sometimes we can see that there is incomplete dominance. And in incomplete dominance, we see that there is something that's intermediate between what we normally think of as the dominant phenotype and the recessive phenotype. We can also have a case of codominance, where both alleles, or many alleles, can be equally likely to be expressed. All right, so here's a case of incomplete dominance using a different flower model. So these are snapdragons. So snapdragons can be true breeding red or white, and in this case, the gene is indicated by this letter C with a superscript, R or W. All right, so true breeding red are CR, CR, and true breeding white are CW, CW. What happens when we cross them? We get all pink flowers in our F1 generation. So we have incomplete dominance. We don't just see all red flowers, we see what looks to be kind of like blending, but it's not really blending for reasons we'll see in a second. So in this F1 generation, we get CR, CW, and the gametes that can be allocated to the progeny will be 1/2 CR and 1/2 CW. So what happens when we do the cross between the flowers in this F1 generation? We end up with our F2 generation having 1/4 red, 1/4 white, and 1/2 pink. In this case, it's the same proportion of offspring, same proportion of alleles being distributed to the offspring, but it just so happens that the heterozygotes are now visible because they're a different color. And this is not truly blending because we only have 3 character states. It's not that we can cross pink and white and get a lighter shade of pink, or cross pink and red and get a darker shade of pink. So this tells us that in the case of dominant and recessive alleles, it's not that the dominant allele is subduing a recessive allele. It's just, we're just talking about the alleles being different forms of genes. And the way that these alleles behave is going to depend a lot at what level we examine the phenotype. It's also a mistake to assume that dominant alleles are always going to be the most abundant within a population. Dominant alleles are not necessarily more common in populations than recessive alleles. For example, polydactyly, there is a dominant gene that leads to offspring having extra digits, fingers or toes. In the US, 1 baby in 400 is born with extra fingers or toes, but typically these children have those extra digits removed shortly after birth. So this is actually a dominant allele. However, just because it's dominant doesn't mean it's going to be the most abundant allele in a population. In fact, it's rather rare. There may also be the case that we have more than 2 alleles for a particular gene. So we actually have 4 phenotypes for our blood groups, A, B, and O. Positive and negative, if you are O positive or O negative or AB positive or AB negative, the negative and positive refer to a different gene, which is called the RH factor. But for just the simple ABO blood typing, we know that there are 3 alleles for a particular enzyme, which we'll call I, which is an enzyme that attaches different carbohydrates to our red blood cells. And there's a carbohydrate A and a carbohydrate B. There is a third allele for this enzyme, which essentially does not encode for any enzyme. And this one is recessive to these 2 alleles, which do place carbohydrates on the red blood cells. So the enzyme encoded by IA adds the A carbohydrate, IB allele adds the B carbohydrate, and the lowercase I, the recessive allele, does not add any carbohydrate. What this means is that we've got 3 different alleles for the gene, which give us 4 possible phenotypes for blood type. We can either have type A blood, which means we've got this A carbohydrate on the surface of our red blood cells. We can have the B carbohydrate, which puts the B carbohydrate on the surface of our red blood cells. You can have both A and B, or you can have none of them and have type O blood. As you can see here, though, there's more than one way that you can be blood type A or B. You can have 2 copies of IB, or 1 copy of IB and a copy of the lowercase I. And the same is true with blood type A. Now, if you have either A or B, if you have both A and B, you're going to express both phenotypes. And if you have neither, you're going to express neither of the phenotypes. So alleles IA and IB are codominant to each other. I'll let you read this at your leisure if you haven't seen it already. Another effect of having a single gene is you can have multiple phenotypic characteristics that develop from a single gene. So one gene can have many effects on the phenotype of somebody who has that particular gene. And that's called pleiotropy. So, for example, in hereditary diseases such as cystic fibrosis and sickle-cell disease, having those diseases means you've got a number of different symptoms. And those symptoms can seem like they're all unrelated to each other. However, it's all going back to one gene. So symptoms of cystic fibrosis may include thick mucus, shortness of breath, sinus infections, pneumonia, growth in the nose, having a foul-smelling stool, being gassy or constipated, inability to gain weight, low bone density, and wide-rounded fingertips and toes. Now, these things seem like they're not necessarily all related to each other, but they do all, we can trace all of these symptoms or these phenotypes back to one gene, a gene for cystic fibrosis. We can trace all of these symptoms to a single allele, to a single allele of a single gene. So many phenotypic effects derive from one genotype is the definition of pleiotropy.